Now we're going to take a look at the second part of protein synthesis. So during transcription, we made a molecule of RNA, and more specifically, we made a molecule of mRNA. Now the mRNA, remember the DNA is stuck in the nucleus, but to make a protein, we need an organelle called a ribosome. And so the mRNA is able to carry the information in the DNA in the nucleus out to the ribosome, which is located in the cytoplasm. For the process of translation to occur, we actually need 3 different types of RNA. One type is the mRNA that we just talked about. And remember that this is just the DNA getting converted into mRNA during that process of transcription. Additionally, we need rRNA and tRNA. rRNA is also known as ribosomal RNA, and it actually helps to make up part of the ribosome. And tRNA stands for transfer RNA, and transfer RNA transfers or carries amino acids to the ribosome. So what happens is that the messenger RNA goes to a ribosome, and the ribosome is kind of like the factory that it can read the messenger RNA, and it helps to bring the transfer RNA into the ribosome, and then it takes the amino acid. And as it takes the amino acids, it starts making this chain of amino acids, which is ultimately going to become the protein. So let's take a look at how that happens on the next several slides. Now, messenger RNA is made up of several of these nucleotides or these bases that are all bonded together. 3 bases or 3 nucleotides in RNA is known as a codon. All right, so this molecule of RNA is made up of these triplets called codons. And each codon, it codes for one amino acid. So you can see we actually have this chart over here that helps us to kind of figure out which codon codes for which amino acid. Now this chart, you don't have to memorize this chart. You just need to be able to use it as a tool. So just like the periodic table, you don't have to memorize all the individual elements and where they are and how many atoms and stuff they have. Instead, if I give you a chart like this, you need to be able to use it to figure out which codon codes for which amino acid. So let's take a look at this codon here. So this GUC that we see right here. So the first letter is going to be G. So that means that our amino acid is going to be in this row on the chart right here because our first letter is G. Our second letter is U. So that means that our amino acid is going to be in this square right here. And that's because U is our second letter, which means that we can take a look at where these two the column and the row overlap. Our third letter, which I kind of messed up, but that's a C right there, is going to be this row right here. So we can find our codon here for GUC, and that translates to our amino acid known as VAL, which is also known as valine. So anytime there is a GUC on an RNA sequence, it's going to code for an amino acid known as valine. Now we can look at this other codon here. So we have CCA. Let's do this one in blue. So on this one, our first letter is our C. So we know that the amino acid is going to be in this row right here. Our second letter is C, so it's going to be in this row right here. So we can look at where the two overlap. So we're narrowing it down. And then our last letter is A, which is the third one down. So you can see CCA codes for PRO, which is the same thing as proline. So now we know, and these are the amino acids over here that I'm writing here. So if we have a sequence of DNA, we can then figure out which, or what the sequence of mRNA is going to be, and then we can figure out what the codons are, and then use this chart to figure out what the amino acid sequence is. So essentially, to kind of break this down, you have DNA that gets converted to mRNA. The ribosome exposes one codon at a time, and each codon gets, or it kind of codes for the location of an amino acid. And a chain of amino acids is what gives you a protein. Now, in addition to this chart coding for the different amino acids that are going to be in a protein, we also have what we call start and stop codons. So the start codon is AUG, and it codes for an amino acid known as methionine. So all proteins start with the amino acid methionine. And the reason why we have to have this start codon is because remember that during our post-transcriptional processing, remember that our molecule of RNA got that poly A tail and that 5' cap added to either end. But these do not need to be translated into a protein. Remember, these are just throwaway nucleotides that are there for protection. And so the ribosome knows to start when it finds the codon of AUG. And so the ribosome is going to come in and expose just that codon there. Now, this translation is going to happen kind of in one direction along the RNA, where the ribosome is now going to expose the next codon and then the next codon. But ultimately, it has to stop, and it has to stop before it gets to the other end of the RNA, because once again, the 5' cap and the poly A tail are not supposed to be translated. And so if the ribosome comes across either UAA, UAG, or UGA, these are known as the stop codons, and it's kind of a signal to the ribosome that it needs to stop the translation process, and it detaches from the RNA, and then the protein is done being made. Once again, you don't have to memorize these codons. Just be able to read the chart and understand how to figure out which codon codes for which amino acid, and be able to know where the start codons are based on what we see in the chart here, and where to stop based on the stop codons here. So recall that the mRNA has a long strand of nucleotides. And these nucleotides, the order of them was determined by the strand of DNA that it was made from. Remember, we've got our guanine, cytosine, adenine, and then instead of thymine, we have uracil. And so we can go ahead and just kind of finish out this strand here by putting some random nucleotides in. Now, remember that 3 mRNA nucleotides is equal to a codon. So, for example, this GCA would be a codon. The UUA would be a second codon. The CCG would be a third codon. And then the A would be the first part of the later codon here. Now the mRNA meets up with the ribosome, and the ribosome actually exposes one codon at a time. So the ribosome is basically helping to kind of expose just one codon at a time. And exposing that codon helps the tRNA be able to kind of meet up with the mRNA. So what happens is that tRNA, our transfer RNA, it has 3 bases attached to it. And these 3 bases are known as an anticodon. So as our ribosome is exposing this codon here, the tRNA that has the matching or complementary anticodon is going to kind of fit together with this codon like a puzzle piece. So for example, this would be C pairing with G, G pairing with C, and U pairing with A. Now this tRNA molecule has an amino acid stuck to it. All right, and remember we can use that chart to figure out which amino acid is stuck to it. So what happens is that the ribosome, so we'll kind of draw like a little dotted line here. So the ribosome comes in, it exposes this codon and allows the tRNA to come in with the matching anticodon. And once that match is made, the ribosome actually takes this amino acid and it holds on to it. I guess I should do that in yellow. Then that ribosome is going to move down the line to expose the next codon. And so another molecule of tRNA is going to come in, and it's got to be complementary to this codon. So in this case, it would be AAU. And this tRNA would have a different amino acid attached to it. So the ribosome, like I said, would move down, and the tRNA would come in, and it would actually drop off its amino acid, and the ribosome would form a bond with the amino acid it got from the first tRNA molecule. So here, I'll label them here as 1 and 2, and 1 and 2. Then the ribosome, is going to move down to expose the next codon, which is going to allow the next tRNA molecule with its anticodon to come in. And it's going to be attached to amino acid number 3. And once it matches, the ribosome is going to take that third amino acid from the tRNA and form a bond with the other amino acids it got from the first 2 codons. And this is going to keep happening until the ribosome hits that stop codon, and then it's going to release the mRNA, and it's going to release this polypeptide up here. So this chain of amino acids that's forming is called a polypeptide. And this is just a chain of amino acids. So you can see in this picture down here, this is showing the mRNA here and the ribosome here. And we've got our tRNA molecules with our anticodons. And we have the ribosome exposing codons one at a time. And you can see how the tRNA molecules are coming into the ribosome. And if the anticodon matches the codon in a complementary way, what happens is that the amino acid gets dropped off at the ribosome. And you can see that the ribosome is going to take that amino acid and form a bond with the last amino acid it had. And so this is going to keep happening until we hit that stop codon further down the messenger RNA molecule. Now remember that the ribosome itself is made up partially of RNA, and it's actually called rRNA, which stands for ribosomal RNA. And it just helps to make up part of the ribosome, and the rest of the ribosome is made up of proteins. And these ribosomes actually have two subunits. They have a large subunit and a small subunit. And these subunits can actually come apart and then come back together depending on their usage when they're being used. Now, the RNA and the proteins are made in the nucleolus. Now, remember, the nucleolus is just this region in the nucleus that codes for these ribosomal subunits. And as we've already established, we need the ribosome in order for translation to occur. And it kind of helps to facilitate or kind of play matchmaker between mRNA and tRNA. It helps to kind of bring the whole process together so that the anticodons can match up with the codons. Now let's do a quick review of amino acids. We talked about amino acids way back at the beginning of the semester when we talked about macromolecules. So remember that proteins are one of our macromolecules and they're one of our polymers. And remember that polymers, so a protein, like we said, it's a macromolecule and it's a polymer. Remember that poly means many. And polymers are just these large molecules that are made up of many monomers bonded together. So remember, mono means one. So amino acids are the individual building blocks that bond together to form this polymer that becomes a protein. Now there are 20 different types of amino acids, but they all have the same basic structure in that they have this central carbon. They have an amino group, which is a nitrogen bonded to two hydrogens. They have a carboxyl group, which is a carbon double bonded to an oxygen and also bonded to an oxygen that's bonded to a hydrogen. They also have this kind of single hydrogen coming off of the central carbon. Now, the key difference between our 20 different types has to do with this kind of what we call the R group or the side chain. So you can look in our chart over here that all of these amino acids have the same basic structure except for the shaded areas here. So some amino acids are really simple. So glycine, instead of having this R, this R is just kind of a placeholder. So instead of having an R there, it would just have a hydrogen atom. But alanine has a carbon bonded to three hydrogens. And you can see that valine is even bigger, and it actually has a little bit of branching going on. You can see that this R chain here, or this side chain, it can be a long branch of carbons and hydrogens. You can see that there are also some nitrogens here. Sometimes there are some sulfurs in this side chain. Sometimes these chains form rings. So this R side group here is just a placeholder for all these different possible combinations. And all of these different combinations, they just allow these amino acids to interact with each other in a way that ends up giving the protein a three-dimensional shape, which we'll talk about that more on a later slide.